To begin your transformation, carefully pipette 10 microliters of your DNA to your cold -sign experimental tube that contains competent cells. Do this by gently pipetting out your DNA while swirling and stirring the solution. Stir for another 15 seconds and then place your cells back on ice for 20 minutes. Use a similar procedure for your positive sign control cells. In this case, add 5 microliters of control DNA. Just like before, gently pipette out the DNA while swirling your pipette. During this incubation, your DNA is being bound up by cations and it's also binding to the cells. While your samples are waiting on ice, get your heat shock water bath ready. Get a big bowl or bucket and use tap water and your thermometer to achieve 42 degrees Celsius water. The water bath should be 42 degrees Celsius for the next step, so make sure to double check the temperature is right before you start. After the samples have been on ice for 20 to 30 minutes, it's time for the heat shock. Take the cells from the ice and dip them into the 42 degrees Celsius water bath for 90 seconds. During the heat shock, the membranes of the cells become semi-permeable to the high concentration of salts and DNA in the environment, and your DNA can enter the bacterial cells. After 90 seconds, it is extremely important to immediately place the cells back on ice for at least 2 minutes. This will make the cell membranes more rigid and trap any DNA that has entered the inside of the cells. Then, it's time to recover your cells. Do this by adding LB media to your tubes. Shake the cells and then place them in the 37 degrees Celsius incubator for 30 minutes. During this incubation, the cells are recovering from the horrendous living conditions you just put them through. Also, the plasmid you transform the cells with start to create the protein that makes the E. coli resistant to chloramphenicol, the antibiotic that is in the LB agar plates. And now is a great time to work on your lab book entries on Synbiota. Just before the 30-minute incubation is complete, you will need to label your LB agar plates. Label 2 of the plates with your names and a negative sign, since these will be for the experimental DNA. Label the 3rd plate with your names and a plus sign, since this plate will be for the positive control. After the 30-minute incubation is complete, pipette 100 microliters of the experimental DNA cells in the minus tube onto an appropriate plate. And then pipette 50 microliters of experimental DNA cells onto the other appropriate plate. The cells can then be spread out using a single sterilized plastic spoon or spreader. Then pipette 50 microliters of positive control DNA cells with the plus sign onto the remaining plate and spread out the cells with a different sterilized spoon. Leave the plates with the lids on top and place them in the incubator for 30 minutes. This will allow the cells and LB media to soak into the agar, as well as allow for some evaporation of the LB media. Once there is no pooling LB media on the surface, the plates can be flipped over and incubated for 24 to 48 hours. The flipping of the plates is important to ensure that the agar and cells remain moist from water vapor overnight. We hope that this has transformed your understanding of how to do transformations well.